Hi, everyone. My name is Elizabeth DuPre. I'm a PhD candidate at McGill University, working with Dr. Jean-Baptiste Pauline, and I'm so delighted to be here as part of the NIMH workshop on naturalistic data and individual differences. And as part of this workshop, I want to tell you a little bit about how we're thinking about the problem of finding similarity across neural diversity. And in particular, I want to talk to you about some of the work that we've done to think about how we can assess the generalizability of individual brain models. So to motivate this work, I just wanted to talk about a really core challenge that we face in the field, which is how do we make comparisons across different research participants? And in particular, the question really is how can we establish a mapping between different participants' neural activity patterns? So for example, if I have activity from participant A and activity from participant B measured using the same modality, how can I say that they have some correspondence between them? For example, that they're in the same functional region or even within the same broader connected functional network. And the traditional way that this is done within the field is to normalize to a standardized anatomical space. So for example, this could be the Montreal Neurological Institute or MNI space. But we know that normalization, standard anatomical normalization, does not fully address the problem of intersubject variability. And this is actually something that we've known for quite a while. And so this is a challenge quite broadly across the brain, but it's particularly a challenge in higher order association cortices where we see a much weaker structure function mapping. And what I mean by that is that the same structure can support a variety of functions within and across participants. So if we're looking at this structure, how do we really know that we're looking at the same function when we look across participants or over time? And obviously, because this is a known problem, we aren't the first to point this out. And this is something that the field has thought about really quite heavily for a long time. And the current consensus around the best way to deal with this is to use standard anatomical normalization and then to do Gaussian smoothing. And Gaussian smoothing is actually useful for a host of reasons, but for the purposes of making comparisons across subject, I just want to point out that Gaussian smoothing is really useful because it helps to quite literally smooth over any remaining intersubject variability after anatomical alignment. So if you've worked with MRI data before, you're likely already familiar with this problem, and you may have a solid mental model of what's really going on here. But I think it's sometimes quite useful to think about these problems from a slightly different angle. And so to do that, I just want to make an analogy to a different domain, which you may be less familiar with, which is the idea of face matching or face morphing. So similarly to MRI data to brains, faces have really consistent structure. So with brain data, we're thinking about the various lobes of the brain, sulci and gyri. With faces, we're thinking about standardized anatomical landmarks. So these are things like the eyes, the nose, the mouth that are really quite consistent across individuals and that we can use to generate a mapping even within an individual. So you could think about distance between the eyes or distance from the bridge of the nose to the mouth, etc. But because these landmarks are so consistent, we can really use them to define our mapping. And we can then actually look across participants. So the former image was a single former US president, President Barack Obama. But we can not only look at Obama's face, we can actually look at a variety of other faces. So here on the left, I have in total 6 former US presidents. And you can imagine, just as I showed you in the last picture, we could go through each of these faces and we could identify the relevant landmarks around their eyes, nose, and mouth, and the contours of the face. What I can then do is to actually take all of these faces and use the identified landmarks to generate a mapping between them and actually put them into a composite or template space. So on the right, I'm showing you what this composite face looks like. And what this face really represents is an average across all the 6 US presidents on the left, kind of the template president, the average president. And there are 2 really important things that you can take away from this. One is that those landmarks that we use to define our mapping within a face, things like the eyes, nose, mouth, those are all really well preserved and really well represented in our template face. So here you can quite clearly see the average eyes, the average nose, the average mouth across these 6 US presidents. What you can also notice is that the more idiosyncratic information that we didn't specifically define in our landmarks, things like the hairline, for example, are not as well represented. And so this tells us that the landmark definition becomes really quite important if we want to make sure that information is carried across. The other thing that you can notice right away is that the composite face, the template face, doesn't actually resemble any of the 6 faces that were used to construct it. And so this is really quite critical because it tells us that while this composite template face is incredibly useful, it tells us really structured information and commonalities across all of the different individuals that were used to construct it. It doesn't allow us to kind of trace back to their idiosyncratic features and make inferences about their individual differences in the same way that we could if we looked at the original images. And this problem is particularly exacerbated for those parts of the face where, again, we didn't include them in our landmarks, so they weren't used to kind of drive the alignment. And so what we can see is that much as in the face, the brain can suffer a similar problem where we're using these anatomical landmarks, and so those will be very well represented, but more idiosyncratic features, particularly functional features, may not be well represented, and it can be difficult to see how to preserve them in this kind of template mapping. So I'm here to suggest that one really promising potential alternative is functional alignment. And why do I think it's a promising alternative? Well, because rather than relying on these predefined anatomical landmarks, we can instead take a landmark-free approach. So rather than saying we're looking for correspondence at specific points, we're going to actually focus on directly mapping functional activation across participants. And I'm going to go into a bit more detail about what that really looks like. But I just want to point out, for the sake of terminology, you may have already heard of this class of methods. You may know them as hyperalignment methods. And the field kind of flits between these two terms of functional alignment and hyperalignment. But this is all really the same body of work that's focusing on this question of how can we directly map participants' functional activation patterns. For the sake of clarity, in this talk, I'm going to be using the term functional alignment, but again, hyperalignment is something you may hear quite often in the literature. Okay, so this sounds great if I want to take this landmark-free approach and focus on aligning participants' functional activations, but how can I really do that? What does that really mean? So if I have a participant's brain, and I have some measurements that I have made using, for example, fMRI, I can then take those measurements and I can sub-select a given, let's say, voxel, and I can do something quite common, which is to look at its activity over time. So here, for example, let's say that this fMRI data was collected while participants were watching a movie. And so I can look at the activity in this voxel in response to every frame in the movie. And similarly, I could just randomly sample another voxel and I could do exactly the same thing. So at this point, there are many different things that could be done. Perhaps kind of the field standard approach would be to do something like functional connectivity, where what you're really doing is you're correlating these two time courses. But there's obviously other ways to think about this data. And the one that's really useful for the purposes of functional alignment is to think about actually defining a new functional space. So here what I mean by that is that whereas before my axes were voxel by time, so voxel 1 by time, voxel 2 by time, now I have this new functional space where my axes correspond to the number of voxels sampled. And obviously, in real data, you'd probably be looking at more than 2 voxels, but for the sake of illustration, let's focus on 2. It just makes it a bit easier to see. So if I have these 2 voxels and I have their time courses, I can just step through the movie. So for example, let's say this is a given frame. And here I see that voxel 1 has relatively high activity and voxel 2 has relatively low activity. And I can just include this as a plot in my new functional space, my new voxel space. And so this point now encodes information about the relative activity of these two voxels at this point in time, at this frame in the movie. And of course, I can just continue to step through the movie and do exactly that. So here, for example, you see the reverse trend. Voxel 1 has low relative activity, voxel 2 has high relative activity. And this is now another point in my functional space. And of course, you can do the same thing. So what I can do is actually step through the entire movie and generate a distribution that maps the functional activity, the relative functional activity of all of these voxels that I'm including. And again, obviously, with more voxels, this would be much harder to visualize, but this is the basic principle of what's happening here. So this sounds great. This is for one participant, however, and you can imagine exactly like this. Here we've stepped now from 2 to 3 voxels, still the same idea of 3 frames in a movie, and we have this one participant space. But if we want to use this to make comparisons of our subjects, we'll obviously need more than one participant. So here, if we have a second participant who's done exactly the same thing, watched a movie in the scanner, we can now use their relative activations in the selected voxels over the course of the movie and plot them just as well. And so what you can actually see right away is a visual correspondence between these two participants. And you can even imagine kind of mentally rotating these different coordinate systems so that they overlap, so that the frames overlap. And here you could say that this is putting them into some common space. And so if you actually wanted to get from one participant to another, you could just take the composite of these transforms. So here, this would take you into this this other participant space, right? And so this is incredibly useful because now we have a method where we have these voxels that don't have any defining anatomical features by which to align them, but we're actually using the functional activation to drive the alignment itself in this landmark-free way. And we can move directly from one participant to another. So this is really exciting and kind of underpins this idea of functional alignment. Now, there are a couple of really important questions that I just want to address right off because this is going to constrain how we think about the problem. And these questions really all kind of center around the concern of how can we make sure we're learning appropriate transformations? When we think about anatomical transformations, we have a bit more information to work with. We have visual information to work with because this is working in the three-dimensional space of the real world in which brains exist. But we're in this high-dimensional functional space. How can we really make sure that we're learning something appropriate if we can't inspect it directly? So there are two kind of main things that we can think about here. One is that we can make sure that we're learning transformations within a local neighborhood. So this is really useful in that it acts both as a form of regularization, so the amount of data that we're working with is reduced, and it also helps prevent biologically implausible transformations. For example, you don't want to align contralateral hemispheres or the visual cortex to prefrontal cortex across participants. That's not something that we would expect to see in most healthy young adult subjects, for example. And so when I say constraining within a local neighborhood, just to be a bit more precise on what I mean, this is something that is obviously experimental defined and question driven, but there are two kind of broad categories perhaps that we can think about. One is what we call piecewise alignment, and this is also sometimes thought about in the context of regions of interest. The basic idea here is that you're learning local neighborhoods that don't overlap. So here, for example, we have a red area, we have a blue area, and we learn two different alignment transformations in each of them. And then if we want to, for example, generate a larger transformation for the combined area, because these two regions don't overlap, we literally just stack our transformations into a larger transformation matrix. And this way, we can both learn our transformations within local neighborhoods, but then apply them to larger contexts, such as the whole brain, for example. Another way to think about defining a local neighborhood is that rather than a priori assuming some kind of functional or anatomical boundary based on, for example, a field standard parcellation, you could instead take a more agnostic approach and use what are known as searchlights. So these are quite popular in the field and really useful in that they allow you to kind of scan across cortex and create smooth representations of underlying function. And the reason they're smooth is that because unlike piecewise alignment, where you have these hard boundaries of non-overlapping regions, in searchlight alignment, you actually do allow, you really enforce overlap between different regions. So here, whereas before red and blue didn't overlap at all, now, although red and blue still don't overlap, we intentionally introduce a third green region, which is the overlap between these two regions. And so what this allows us to do is to have kind of a smooth representation across cortex without any a priori boundaries. But it does mean that if you then want to aggregate these transforms, so if you want to look across the composite area, you now have overlapping transformations. And so you need some way to similarly aggregate the overlapping transformations. There are lots of ways that you could do this, but here in this figure, what I'm showing is you just average where the overlap occurs. And these two cases, because we define our local neighborhoods slightly differently, results in different properties for our transformations. But in either case, they are providing some kind of local neighborhood context, which is going to constrain the alignment and prevent biologically implausible transformations. So they're both really useful in this context. Okay, so we have various ways through which we could learn a local neighborhood, and that will help us to make sure that we're learning appropriate transformations. The other thing is that we want to make sure that these transformations robustly map to individual functional spaces, because if the transforms are appropriate, they should enable us to be able to map to an individual space and to do so in a way that preserves information. And so this is something we're going to focus on in depth, is how do we assess whether information is preserved. But the first thing I want to focus on is just this question of robust. So the problem, of course, is that if we want to be sure that something is robust, we need a lot of data. We need a lot of data in this case, both within a participant. So we need to be able to really rigorously kind of sample their functional space and have information on their activation patterns in response to, say, a movie or a task or both. But we also need a lot of data from many different participants, perhaps across different data sets or different scanner acquisitions. And this is a pretty big ask, right? So if you want to collect a really large, well-sampled data set, this is quite realistically years of work. And so the work that we're going to talk about today is really only possible because within the field, over the last few years, there's been more and more availability of well-sampled individuals in publicly available data sets. So this initiative was really sort of kicked off with the MyConnectome project by Russ Poldrax and colleagues. And the idea basically is that there's been a shift towards focusing on deep data or densely sampled data, which extensively characterizes a small number of individuals. And as more and more of these data sets have come online, analyses such as the one I'm going to talk to you about today have become more and more feasible. And so this is really kind of crucial to all the work that we've done, is just the availability of these really wonderful public resources. So here on the right, I'm just showing you a figure that's adapted from Naturalistic and colleagues. And the basic idea is to kind of contrast these field standard data sets that you may be familiar with, like the Human Connectome project and the UK Biobank, with along the bottom axis, this idea of densely sampled or deep phenotyping data, which you can see there have become more and more data sets within this realm available just within the last few years. And even some of the data sets mentioned in this figure have been really critical to the work that I'm going to describe. So it's really this availability that makes all this work possible. So just a brief thank you to everyone who's made their data available, because it's so critical here. Okay, so with this context, with this understanding of functional alignment, and with the public availability of these really richly sampled data sets, my co-authors and I, in particular, the co-first author on the work I'm going to talk about, Thomas Basile, and I sat down and we asked, how can we use this data to learn more about appropriate functional alignment transformations and how useful they can be in preserving individual specific information for downstream tasks. So that's a very broad question. So we wanted to ask a few more specific ones. First, we wanted to ask what set of transformation is best to align functional spaces across subjects. So previously, when I showed you the transformations that you could almost imagine mentally rotating them, of course, I told you that as you go to higher dimensions, we no longer have access to that kind of visual check. So there are, in fact, many, many different ways that you can actually think about aligning these different spaces. And so we need to ask, if we're going to use different kinds of transformations, in particular ones that are hard to visualize, which perform best in some context. And to slightly constrain the problem, we're really going to focus here on the spaces that I've shown you, those spaces that are defined by activation in response to, say, a movie, rather than defined by other metrics such as functional connectivity. There are other investigators who've been working quite hard on thinking about using metrics such as connectivity, and that can be incredibly useful depending on your use case. But the way you define the spaces and the way you interpret them is slightly different. So that's not something we're going to touch on here. So once we ask what set of transformations are useful, we also want to ask, are there some contexts where alignment is more useful than other contexts? And there are, again, lots of ways that you can think about this. Kind of as a first pass, what we wanted to ask is, in a whole brain, so where you're aggregating across many local neighborhoods, or in a single task-specific region of interest, if we define alignment at these two different scales, are we going to be able to see that our alignments are more or less useful for downstream tasks? And then finally, I've told you again and again that it's quite difficult to visualize these transformations. High dimensional spaces are just not something that we really have access to. And so we need to find another way to think about what these transformations are really doing. And one way is just to see visually, how do the transformations change the signal? So if we can't look at the transformations themselves, we can look at the signal before and after alignment, and we can get a qualitative sense of how these transformations are altering our signal. And so that's something we really wanted to think about. How can we do that as well? So our goals for this particular study were first to try and quantitatively compare various functional alignment methods at a whole brain scale, so where we're aggregating across many local neighborhoods. Then to quantitatively compare functional alignment methods for task-specific individual regions of interest. This is where we define a region of interest and we just calculate the transform across the entire region, so we don't aggregate within any neighborhoods within it. And then finally, we want to qualitatively compare the transformations generated by each functional alignment method in terms of how they impact our signal. So if we compare the signal before and after alignment, what do we see? And so this sounds like a tall order maybe, but perhaps the hardest piece is just this question of how do you quantify functional alignment? So one way to think about alignment, in fact, is as a reconstruction problem. So maybe what we'd like to do is that we have participants' maps, for example, when they're engaging in a particular cognitive task. And we learn a transformation, and then we see if we apply that transformation, how well can we reconstruct the held out maps? And so if we take this kind of procedure, one thing you could immediately imagine doing is to directly compare the learnt and the held out maps using metrics such as, for example, correlation. So just looking at the distance between them. And while this is really useful, if we want to make comparisons across data sets, which I told you we're kind of interested in this question of robustness, so we want to look across data sets and take advantage of those publicly available resources, we know that Some of these image-based statistics, such as correlation, can be sensitive to low-level image characteristics. So if different data sets have different intrinsic amounts of smoothing, for example, it may confound our estimates. And so it may be difficult to compare across data sets in a rigorous way if we're using these image-based statistics to directly compare learned and held out maps. Alternatively, what we thought to use is actually a predictive framework. And the predictive framework that we settled on is known as inter-subject decoding. So if you're not familiar with inter-subject decoding, I'm just going to give the whirlwind tour. The basic idea is this. We'd like to see how much a learned model generalizes across subjects. And this is a notoriously hard problem because we know, again, this idea of intersubject variability, that we can learn models really well within a subject, but if we want to learn models across subjects, it can be quite difficult because perhaps they have different underlying functional organization that's going to make transferring the model difficult. So what intersubject decoding does to test this is it actually takes all but one subject and you train, in our case, a linear classifier, a linear support vector classifier. And what we're doing is we're distinguishing, in this toy example, 3 category labels. So if we have different beta maps, let's say, derived from a GLM for face, body, and tools, and we have all but one subject, and we have those beta maps, we can learn in this high-dimensional voxel space a linear classifier that distinguishes between our 3 data types. Now, once we've learned this linear classifier, we can actually apply it directly to our held-out subject, and we can assess how well it performs. So in this case, you can see that tools and body are misclassified, but face is correctly classified. So here we'd have an accuracy of 33%. And what intersubject decoding does is just repeat this procedure where we hold out a different subject from our sample every time, and then we look on average how well do these learned models generalize across our subjects. So if we're going to use this predictive framework, this provides a really nice way to kind of quantify how well our alignment methods perform in terms of what is the accuracy of our model. So the next question is, what alignment methods are we going to consider? What are we going to actually look at here? And we actually looked at 5 different unique alignment methods that have all been used in the literature to date. The first is piecewise procrustes, followed by searchlight procrustes. And again, although these are both procrustes-based methods, so they're using the same rotation-based approach to align the different functional spaces, what differs between them is how we aggregate across the local neighborhoods. So as before, it's the question of whether or not we have non-overlapping or overlapping neighborhoods. In addition to these 2 procrustes-based methods, we also used optimal transport, which is another method which is less sensitive to high variance voxels and so may provide a slightly more robust mapping. We also used a latent factor model known as the shared response model, which is quite useful in that it provides an initial decomposition before doing the alignment. So it looks for some latent factors that are shared across participants, perhaps. And then finally, we did a method which is an extension of something introduced by Tabor and colleagues that we benchmarked here, known as intra-subject alignment. And so here you can kind of see what's the difference between intra-subject alignment and all the other alignment methods I considered. And it comes to the direction of these arrows. So here, if you look, the red arrows are the top 4 methods, in that what you're doing is you're learning a transformation within your alignment data, and then you're applying it to some unrelated task data. So here, for example, we could learn a transformation on 2 participants watching a movie, and then we have, as I showed you before, let's say the beta maps for face, body, tools. We then apply that learned transformation there. Intra-subject alignment is different in that what you're actually doing is you're learning a transformation within subjects across different task modalities. So I would learn a transformation from my subject's movie watching to their task data. and then I would take the composite transformation and apply it on my held out subject. And so this is sort of an interesting spin on the idea of alignment, in that you're still aligning, but you're aligning now within subjects between task modalities rather than across subjects within task modalities. So all 5 of these methods, what we are interested in is we're interested in looking at them at both the whole brain, so aggregating different local neighborhoods, and at the ROI, the unaggregated whole task relevant ROI level of analysis. And in every case, what we're going to be doing is we're going to be deriving our estimates of inter-subject decoding accuracy, and we need a baseline to which to compare all of these things. And so the baseline that we're going to be using is standard anatomical only alignment. So this is just the alignment that you might see in something like fMRI prep, where you have a standardized linear anatomical normalization. So our general experimental procedure then will be, first, we'll actually calculate our baseline. So first we calculate our inter-subject decoding accuracy using standard anatomical only alignment. So there's no functional alignment here. We're just using kind of current field standard methods. Then we'll calculate each of our functional alignment transformations. So we'll calculate, for example, piecewise progrestes or SRM. And with the exception of searchlight progrestes, these are generally regularized through piecewise alignment. So what that means is that for each of these methods, we're going to be using non-overlapping neighborhoods. And unless otherwise specified, we're going to rely on the Schaefer parcellation with the 300 atlas resolution. And this allows us to relatively easily make comparisons across methods. And what we're going to actually do then is, if we calculate the functional alignment transforms, we're then going to apply the functional alignment transforms and recalculate our inter-subject decoding accuracy. And we can compare it directly to our baseline to see how does inter-subject decoding accuracy improve or decrease or change, at least, relative to anatomical only alignment. So just to kind of review the experimental procedure, including functional alignment now, we have this figure which I showed you before, slightly rearranged. So again, in panel A, we have some beta maps that we've derived from a general linear model. In panel B, we're training a linear support vector classifier on these conditions using all but one subject. And in panel C, we have a held out subject. This particular subject is held out just for this cross-validation fold. And of course, we'd repeat this for each cross-validation fold. So we hold out all the subjects. But what we're going to do then is we're going to take this as our baseline, and we're then going to learn a functional alignment transformation. So we're going to use this, learn this on unrelated data, let's say on movie watching data. And here what we're doing is we have these 2 activation profiles for 2 different participants. And again, because this is in 2-dimensional space, you can sort of imagine visually rotating it, but there are many different ways that we're learning these transformations, 5 different ways we're going to learn these transformations. And then you can apply this learned transformation directly back to the classifier learned in panel B and all the data you learned in panel B, and then you can retrain your classifier. And so what you're going to have now is a new classifier across subjects. And then you could take this classifier and directly apply it to your held out subject and see, for this cross-validation fold, what's your accuracy now that you've learned this functional alignment transformation. And so, okay, so this is our general experimental procedure and how we're going to assess or quantify, rather, the impact of functional alignment. Okay, so if we use these data sets and our previously discussed experimental procedure, what do we find? So first, looking at the whole brain level, I just want to tell you a little bit about how to interpret these graphs. So what you're seeing along the y-axis is each of the different methods, And then along the x-axis, what's depicted is the relative accuracy change from anatomical only alignment. So here 0% would be that for a given cross-validation fold, you get exactly the same intersubject decoding accuracy as anatomical only alignment. Every dot depicted is a different cross-validation fold, and the dots are colored according to the data set that they come from, as depicted in this legend here. So what you can see, putting all of this together, is that the piecewise shared response model is the best performing method at the whole brain scale. So this is, again, piecewise aggregation, non-overlapping neighborhoods. And this is actually an extension of the shared response model that we specifically introduced in this benchmark. So this was something that was really exciting to see. But of course, there are other methods that also perform quite well. So piecewise optimal transport performs similarly, as does piecewise progressives. And in general, what we're seeing is that the best performing method gives almost a 5% accuracy increase on average compared to standard anatomical only alignment. And this is something that's particularly exciting because if we look at within subject decoding accuracy, so if we just learn and apply our classifier on different subjects, and we compare that to intersubject decoding accuracy with anatomical alignment, we can see kind of a range of values. So we can see how much does our accuracy decrease when we're learning across subjects as compared to within a subject. And in that context, we see about a 10% decrease, a little less, about 8.5. And so this 5% accuracy gain is really encouraging because it's telling us that the best performing method is recovering over half of the accuracy that's normally lost to standard anatomical only alignment can be recovered with this functional alignment method. So this is really, really encouraging to see. The other thing that we wanted to look at for all of these methods was how computationally costly it is to actually calculate them. So these methods, because we're working in high dimensional spaces and because we're working with a lot of data, particularly these longer sequences from things like movies, these can be really difficult to calculate, and we wanted to get a handle on, is this something that most experimenters will have access to? And so what we did is we looked at the relative computation time across methods. So here we're using piecewise progressives as our baseline, which is why it's all centered around 1. And the reason we chose piecewise progressives as a baseline is that it's one of the few methods considered here, really the only method considered here, that doesn't require hyperparameter tuning. One thing to note is that the piecewise shared response model seems to perform quite similarly to piecewise progressives, but we actually did the hyperparameter tuning in a separate supplemental analysis. So if we had in fact been looking for the ideal hyperparameters for the piecewise shared response model directly in this analysis, we would expect it to have a much larger computational time. Otherwise, you can see that many of these methods take significantly longer than piecewise progressives, some 10 or even over 10 times longer than piecewise progressives to compute, which makes them a bit more inaccessible to most researchers. But of course, this is likely to change as new and improved implementations of all of these methods become available. So this is definitely something to keep an eye on if you're thinking about applying these methods in your own work as well. So if this is our pattern of responses at the whole brain level, we also wanted to look and see how does this differ at the region of interest level. So again, at the whole brain level, we're learning inside many local neighborhoods and aggregating those alignments. At the region of interest level, we're learning a single alignment across the entire region of interest, and we don't use any aggregation. So here, first, we're defining task relevant regions of interest, and you can see these depicted in the bottom right. And they're colored according to the particular data set that they correspond to. So for example, Courtois neuromod, which is colored in red, you can see here is defined as a visual region of interest. And this is because in Courtois neuromod, we're decoding a visual working memory task. So we presume that the visual cortex will have some task relevant information. So what we can do then is we learn functional alignment transformations in each of these regions of interest and use exactly the same experimental procedure as before. And so here you can see the results from that. The structure is exactly the same as I showed you at the whole brain. What is different, though, is that because we're no longer aggregating these transformations across local neighborhoods, we no longer have a distinction between, say, Searchlight and Piecewise Progrestes, and we no longer need to clarify that shared response model or optimal transport are performed piecewise. Instead, we're able to just say these methods are learned over the entire ROI. And what we can see then is a very similar pattern to what we saw before, but with some slight differences. So in particular, we see that optimal transport is performing better now than the shared response model on average, although again, there is a relatively large amount of variability across data sets. And in particular, one thing that we found for the shared response model is that one data set, it was really quite difficult to find a particular hyperparameters. So here, the dimensionality that you're going to reduce your data to for the shared response model, for the latent factor model, it was hard to find an effective dimensionality. And so because of the difficulty of finding that effective dimensionality, we saw that the shared response model performance decreased overall because it did not perform as well on this one data set. And so this really speaks to kind of the difficulty of hyperparameter tuning with these models and the need for really rigorous evaluation frameworks in which to compare them and to have a better estimate of their usage in your own data sets. So here then, given that difficulty in finding the effective dimensionality, we see that optimal transport, which doesn't require this kind of dimensionality estimate, is performing better on average. But all the methods surveyed are on average showing some amount of increase compared to standard anatomical only alignment. That is, they're recovering some amount of individual variability that's normally lost in anatomical only alignment. And in this case, again, the optimal transport is the best performing method. We see about almost a 5% increase, which again, in this context is quite significant in recovering otherwise lost inter-subject variability. And so again, on the right, you can see the relative computation time for each of these methods. And we see that as before, we use not piecewise progrestes, just progrestes as our baseline because it doesn't require hyperparameter setting. And you see that the shared response model, where we had a priori done the hyperparameter setting, is very, very fast, and that optimal transport is quite slow, and intra-subject is closer to progrestes. So this is another facet to keep in mind when you want to apply this in your own experimental data. Finally, so we had these two different quantitative evaluations. We wanted to look at the whole brain and at the region of interest, but we also wanted to do a qualitative evaluation because, again, we can't really look at these transformations to see what the transformations themselves look like, but we can look at the signal to see how is the signal qualitatively changing after applying the transformations. And so here what I'm showing you is a participant from the Individual Brain Training Project, their contrast to two different tasks, the two different decoding tasks. So we have this sounds auditory task, we have an RSVP rapid serial visual presentation language task, and we have different contrasts, two different contrasts from each of these tasks. And this is what this target individual's contrast maps look like directly from the GLM. And so what we can then say is if we align every other subject to this target subject, so we're trying to map all subjects into this space one by one, into this participant space one by one, and then we can do a conjunction analysis. So we look to see where there's shared activity across the mapped participants. We can see what the conjunction analysis reveals and how similar it is to the target subject's activation. So how successfully are you able to map other subjects into this one target subject's functional space? So if we look at that for all of our different considered methods, you can see the results here. So here at the top, we have just standard anatomical only alignment. So what I mean is that we just take every subject and we do a conjunction of their activation, and we see how much does that match onto our target individual. Because again, they've already been anatomically normalized to the same space. So this is kind of the field standard approach of how we do correspondence. And we do see that there is correspondence. The field standard works for a reason. But we also see that the idiosyncratic features aren't necessarily well represented, and there's a kind of smoothness to the activations that doesn't exist in the original subject's contrast. So for example, here you can see that this is quite well defined, whereas in the group average, there's kind of blurring over this sulcus. So then if we look at other methods, we can see how well they're preserving these features. So in particular, I want to focus on piecewise optimal transport and piecewise SRM, because these were two methods that performed very, very well. And so what we can see is that these methods do a much better job of kind of preserving the fine-grained features of the target individual's contrasts. So we can see kind of the peaks correspond quite nicely to what was in the target individual's contrast. And so this suggests that these methods are in fact doing a good job at preserving this information, and they're not doing so merely by smoothing. So we're not just seeing, kind of as we did with anatomical alignment, that the correspondence is really coming from this non-optimal smoothing that's blurring the activation. Instead, we see that the peaks of the activation that are defined in the target individual are relatively well represented after well-performing functional alignment methods, which is really encouraging and suggests that indeed this information is not only useful in downstream tasks, but seems from a qualitative comparison to really be preserved compared to the initial estimates. So it suggests, in addition to our other evidence, that we are somewhat successfully mapping into individual functional spaces. So given all these results, we can revisit our goals for this series of experiments and see how we were able to address them. Our first aim was to quantitatively compare functional alignment methods at a whole brain scale. And we were able to do so using this intersubject decoding framework to compare the 5 different alignment methods. And what we see is that piecewise methods perform quite well overall. In particular, we see that this piecewise shared response model, as well as piecewise optimal transport, recover almost half of the variability normally lost to standard anatomical alignment. Piecewise Procrustes also performs quite well, but at a significantly reduced computational cost. So that's really valuable to know. We then wanted to see what would be a quantitative comparison of functional alignment methods for individual task-specific regions of interest to give us an estimate of how different contexts affect the functional alignment methods. And what we find is here, optimal transport performs best. The shared response model also performs quite well, but it struggled to find an effective dimensionality for some of the included data sets, pointing to the importance both of selecting appropriate hyperparameters and also having really rigorous evaluation frameworks for these different methods. And then finally, we wanted to qualitatively compare the effects of these different functional alignment transformations to see how they were changing our signal, given that we couldn't look at the transformations themselves. And so what we find is that the best performing methods are retaining signal specificity. So we aren't just seeing these effects due to some kind of smoothing, isotropic smoothing. Instead, we're seeing that idiosyncratic functional activations are relatively well preserved when we map other subjects into one subject's functional space. And so this is really encouraging and suggests that indeed functional alignment may be one way to deal with making comparisons across subjects more directly in their own functional spaces. So there's a lot of really exciting directions in this area. Some of the ones that we're thinking about right now are to first examine the impact of additional task contexts. So here, all data sets except IBC only had one decoding task and IBC had two. But of course, you can imagine that there may be other task contexts where this alignment has different implications. And so this is something that we're really excited and working on right now. Another thing that you can imagine is because we have this wide variety of available data sets, currently we're only thinking about how can you make comparisons across subjects within a given data set, but it would be really interesting to think about, is there a means through which you can make comparisons to subjects across data sets in the same way that we can make comparisons across data sets normalized to a similar anatomical space. And then finally, something that I know many people are thinking really hard about already is considering how these findings apply in different developmental and clinical populations. And in particular, for different developmental populations, I just want to plug that I know Dr. Tamara Vanderwall has a talk in this same workshop series where she'll be thinking about developmental fMRI, using naturalistic stimuli in that context. And she's already started to investigate and to think about how can functional alignment methods be used in developmental contexts. So if you're at all interested in this, I'd really encourage you to check out her talk and her work. So with that, I just want to say thank you to my supervisor, Jean-Baptiste Pauline, my lab, Thomas Basile, Bertrand Thirion, the Individual Brain Charting Project, which has been so instrumental in this work, Pierre Bellec and Courtois Neuromod, which has also been a huge driving force in all of the work I've done around deep phenotyping and functional alignment to date. Obviously, all of my funders, so much for their support, and you for your attention. And with that, I just want to leave you with a few take-home ideas and a link to the code in case you're interested in checking out these methods yourself. So thanks so much.